Good morning. My name is Nicholas, and today I'll be discussing my research titled The Assessment of Gene Therapy for Heart Cell Regeneration in Mammals. Basically, that means fixing your heart using gene therapy. First of all, I would like to thank my mentor, Dr. Hakan Koskin from Harvard Medical School, for guiding me through this process. And my objective was actually to create a literature review that provided a meta-analysis of the current landscape for gene therapy as an avenue for cardiovascular diseases, namely heart failure. So let's start off with why heart failure and what is it. So heart failure is a lifelong condition where the heart muscle doesn't pump sufficient blood for your body's needs, and it usually occurs if the heart muscle is too stiff or too weak. And why is it significant? So it accounts for 8.5% of all heart disease deaths in the United States and contributes to 36% of all cardiovascular disease deaths. It's currently estimated that the 5-year mortality rate for heart failure is 50%, which is much higher than some other cancers. In total, heart failure kills around 17.9 million people each year, and it is the primary cause of death worldwide. And why is there a need for the development of novel therapies? Currently, there are a wide range of surgical and pharmacological treatments available, yet the morbidity rate due to heart diseases remains unacceptably high. Heart transplants are seen as the gold standard for the treatment of heart failure. However, the supply of organ donors is unable to meet the large demand. And novel treatment methods, such as gene therapy, hold promise in providing revolutionary new methods that have the potential to greatly decrease the mortality rate of patients. So what are the current targets for gene therapy? So right now, the focus is on reactivating cardiomyocyte proliferation. So that basically means that your heart cells can regenerate themselves and renew themselves. Since the adult mammalian heart cannot regenerate itself when it is injured, it results in cell death. So that means once you've injured your heart, you die. Now, another target is to manipulate the different gene therapeutic targets. So here we have transcription factors, miRNA. So mRNA stands for microRNAs. So they're small, non-coding RNAs which help regulate gene expression. And you have siRNAs, which are small interfering RNAs, which can be helped to use silence genes in treatment. And the difference is that miRNAs are highly specific with only one mRNA target, whereas the latter can have multiple targets. And we'll dive deeper into the different techniques later. So the current treatment methods, as I said earlier, you have pharmacological and non-pharmacological treatment. So examples of device therapy would be cardiac resynchronization therapy and mechanical circulatory support. And obviously those come with a lot of limitations. And you have drugs that target the neurohormonal system, such as beta blockers or angiotensin-converting enzyme inhibitors, that have highlighted the limitations of current pharmaceutical methods and have led to a low actual percentage reduction. Overall, there's clear need for improvement here. The success rates are very low, and there is a clear need for change in the field of treatment for heart failure. Surgical strategies to combat heart failure are risky and come with a host of operative and post-operative complications, such as the risk of death, stroke, and infection. Surgery is also very dependent on the quality of viable myocardium that the procedure can improve. So what is the overall aim of gene therapy? As I said earlier, to restart cardiomyocyte proliferation and thus causing it to re-enter the cell cycle. So basically when you're a baby, your cardiomyocytes or your heart cells can proliferate and regenerate, so that grows your heart tissue. But as they mature, they lose this ability. And this means that when your heart is in the neonatal stage, it results in an exponential growth of new tissue as each cell divides to form 2 identical daughter cells. And gene therapy aims to transfer potential therapeutic genetic information to existing cardiomyocytes in the heart, so when you're an adult, to modify the expression and levels of proteins or transcription factors in order to restart cardiomyocyte proliferation. This prevents the formation of scar tissue, and thus allowing adult cardiomyocytes to regenerate and form healthy, functioning tissue, allowing recovery from impaired cardiac function. So why is the cell cycle suspended after maturity? It sounds like having your heart cells having the ability to regenerate would be positive, but once you grow older and you become, well, and once you're born, the transition to aerobic respiration and change to external nutrition results in mitochondria maturation, leading to increased production of reactive oxygen species, and thus induces a DNA damage response pathway involving histone modifications, which basically means it doesn't work anymore. And the metabolic rates increase to maintain temperature and circadian rhythm, which also contributes to the cell cycle being suspended. Yeah, and this is just a quick diagram of the cell cycle. So current methods for gene therapy involve gene replacement therapy and gene editing. So it's exactly what it sounds like. Gene replacement therapy is just changing out a gene, replacing it with a new one. Gene editing is just changing it so it works. So let's talk more about gene replacement therapy. So that involves inserting genes into the target cell in order to control or change how the current genes are expressed. So this requires a vector. So think of it like a delivery van. To deliver the functional copy of the transgene to replace the defective gene within the cell and allowing the functional protein to be produced. So these vectors can be viral or non-viral based, and different transgenes will have different impacts. So these are the current angiogenic growth factors of interest. I won't dive too deep, but this is just a few examples. And they all have different impacts on cardiomyocytes. So the cell cycle is a potential therapeutic target. Like I said earlier, this is just a quick thing for you to read in case you didn't get it just now. I'll talk about how it's regulated. So you have several transcription factors and miRNAs. So there are two main types of miRNAs. You have inhibitor and proliferative. So the stimulation of specific miRNA pathways and expression of particular transcription factors have been found to lead to an increase in the capacity for cardiomyocyte renewal through animal studies. And there are also multiple signaling pathways important in regulating the proliferation of cardiomyocytes, such as fibroblast growth factor 10, found to promote cardiomyocyte renewal or have a significant role in its regulation. And actually, the knockout or inhibition of other signaling pathways can lead to the promotion of cardiomyocyte renewal. So how will we target the cell cycle? Through the use of mRNA and transcription factors. And this is a diagram I created that shows the different impacts of different transcription factors, signaling pathways, and miRNAs. So as you can see, they have almost a double effect. So different ones will inhibit and others will lead to proliferation. So the three main tools being currently researched are siRNAs, mRNAs, and CRISPR-Cas9, which wasn't mentioned previously. So siRNAs are transfected using viruses or liposomes that result in a degradation of target mRNA. So the gene coded for in the degraded section is no longer expressed. So basically it gets rid of what you don't want. And miRNAs are an alternative method for gene therapy, which can also cause mRNA degradation, eliminating the mRNA and repressing the cell cycle. So these kind of have, that example just now would be a silencer. And an example of another potential technique are adenovirus and adeno-associated viruses. So these are one of the most promising vector platforms for in vivo gene transfer due to their unique advantages, including their wide host cell range, relatively high transduction efficiency, low immunogenicity, and the ability to integrate stably at specific sites in the host genome to provide a long-lasting transgene expression. So these are the clinical trials I looked at during my research. Clinical trial 1. So this was carried out to determine whether gene transfer using a viral vector could be safely administered to patients with congestive heart failure and whether it would prove to be effective in its treatment. Completed in 2017. Unfortunately, there were no results posted, but this shows that there are lots of research being carried out. Clinical trial 2. So this is an interventional study that focused on the efficacy and safety of genetically targeted enzyme replacement therapy for advanced heart failure. So the safety of the gene transfer agent MIDCAR was evaluated, and its ability to improve heart function with congenital heart disease was assessed. So results showed that 22.2% of those who had been administered with the high dose of MIDCAR showed signs of improving their heart failure. Whereas 50% of those administered with the placebo or low-dose MIDCAR showed worsening heart failure. 14% of those not administered at all were hospitalized due to heart failure-related issues, while 7.1% had a fatal cardiovascular event. So this shows that the genetically targeted enzyme replacement therapy used did have some positive impacts. So the range of studies that are currently focused on the AAVs, mentioned before, cardiomyocyte division and miRNAs. Yes. So let's move on to the discussion. So like I said earlier, different treatment methods. We should focus on the novel treatment options here, as gene therapy is one of the main novel treatment options. So it's undergone significant advancements in recent years, and recent preclinical trials show encouraging results regarding cell proliferation in small animal models. Another one would be stem cell therapy, which most of you might have heard of. However, it has low efficiency, differentiating into cardiomyocytes, despite the promising idea of implanting new cells into damaged myocardial tissue. And there have been no types of stem cells that meet the ideal requirements for a stem cell to be used in a clinical trial, and there's no guarantee that they'll be able to integrate properly with existing heart cells and heal you, basically. Now you have the key limitations of gene therapy. So there are key physiological differences between humans and small animals, which could negate the relevance of many of the preclinical trials. Current animal models do not fully reproduce the effects of the disease on the human heart, and do not wholly recapitulate the etiology of chronic heart failure. Gene therapy is a highly novel option, and there are many unanswered questions and gaps in our knowledge. And there's yet to be a therapeutic method that uses gene therapy, which has a noticeable impact on the patient's condition. To conclude, so cell regeneration via gene therapy is highly plausible. However, more research and trials need to be conducted to further our understanding of gene therapy, its mechanisms and effects on cardiomyocytes. Clearly, right now, there is no working therapeutic method that uses gene therapy. So for those of you that are wiser in the crowd, I wouldn't hold your breath for it, but maybe for those of us that are younger, maybe 80 years from now, we could have this instead of open heart surgery. Many questions need to be answered. For example, what other kinds of genes need to be targeted, the most effective method of treatment and delivery, gene therapy and its role in heart tissue regulation, and sort of just more research to determine the effectiveness and validity of gene therapy across a greater range of patients. And just as a comment, current ongoing clinical trials that use gene therapy as a potential therapeutic target are correctly focused on targeting cardiomyocytes. So there is a lot of potential here. Thank you.
Thank you so much for giving us a well-compiled presentation. Any questions from the audience? Yes, please.
So when you said about DNA modification or replacement, is the name that translated to the microbe or next dimension?
Yeah. So basically, like I said earlier, you have gene replacement and you also have gene editing. So if I interpret your question correctly, it's whether it's like you're putting the correct gene into the cell. Is that right? Yeah. So that would be gene replacement therapy. So you would take out the damaged gene, or like the gene that's currently suppressing the cell cycle, and then you'd insert the gene that would be created, for example, in a lab that you would want, that's found in the neonatal cardiomyocytes, and then you'd basically stick that in the cell, and then hopefully it would work. Yeah, and then that would cause the cell cycle to sort of restart itself. Yeah.